Yes. So it's going to complex of CDK4 and 6, and now what does that do? Now you have your active complex. Now what is it going to do? Once it's bound, what's its claim to fame once it's bound?
Phosphorylates RB.
Yes, so it's going to partially phosphorylate, so it's going to phosphorylate some of those residues on RB, and that's going to do what?
That releases E2F.
Yeah.
And allows transcription of cyclin E.
Yes, and now what's going to happen?
So then when cyclin E is made, then it complexes with CDK2.
Yes.
That further phosphorylates RB protein.
Yes.
And now that RB is, I guess, fully off of E2F,
Yes.
now you can transcribe cyclin A.
Right, and that's what's going to get you into. You need cyclin A. Do y'all remember that figure? This figure here, you need cyclin A to get you into this phase. So cyclin A now is going to get transcribed, and that's what's going to get you into S phase. So that's the transition from G1 to S phase. Is that full relief of E2F? Okay. All right. Okay, who wants to take a shot at how a protein block can tell us something about the cell cycle? Green shirt, yes, with UTD shirt on. So how would a Western blot be able to tell us? Right, he knows. He's thinking. Do you know what a protein blot is? A Western blot. So what information can you get from a Western blot about the cell cycle? Just what can you learn? Right, exactly. So like in that one slide that I keep showing over and over again, we know that platelet-derived growth factor leads to what? The production of what? Cyclin D protein, right. So you can get information about what proteins are made, like you said, at what particular time of the cell cycle by looking at a Western blot. Okay. Luciferase assay. Who wants to take a shot at that? Okay. What does luciferase tell us?
You can take a promoter and attach it to the luciferase gene.
Mm-hmm.
Then you can actually see what kind of CDK or whatever else will induce transcription of luciferase. And depending on what CDK is active and what cyclins are present, you can see, what would transcribe luciferase. And depending on the fluorescence of the solution afterwards, you can see, is that active? Is that causing transcription?
Right. And even the other part too is if it's getting inhibited, because there's going to be inhibitors of the cell cycle as well. Yes, thank you. Immunoprecipitation? Yes.
You can have like an antibody that binds to like one particular protein and protein complex, and then take out the remnants from the supernatant, and you can find like these things complex in solution, these things like are they present in the cell and in what amount, because you kind of combine with Western blot.
Yes. What about the in vitro kinase assay? Glasses, back. Yes. In vitro kinase assay, yes.
Determine if proteins are complexing with proteins that inhibits cell cycle progression.
But what's the important here? So the important part here is that kinase part.
Yeah.
So what is it telling us?
If the complex is being phosphorylated or not, you have to keep the complex together or not.
So with the in vitro, what that's really sort of important for in terms of cell cycle is determining if those CDK cyclin A complexes are actually active. And someone has said in the green shirt, UTD shirt, was talking about different times of the cell cycle. So one of the things we know is that these cyclin CDK complexes are only active during certain times of the cell cycle. So that's the other thing you can do with that in vitro kinase assay is figure out when something is or isn't active, because it's not always going to be active. Okay. What experiments could be done? This one's a loaded question. What experiments could be done with FACS cells that would provide insight into the cells? So remember, the FACS cells are the ones that are actually sorted, and you collect them to go back into the lab to do something else with them. And that alludes to UTD shirt's statement. So actually, I'll have you answer it. So fast cells, how could that provide insight into the cell cycle? It has to do with timing, what you were saying about timing. So remember, let me give you an idea here. So remember, when we do flow cytometry for the cell cycle, remember we were propydium iodide staining those cells to measure the DNA content? And so we could decide which cells were in G0, G1, we could determine which cells were in S phase, and we could determine which cells were in G2M phase. So what if you sorted based on cell cycle phase and collected everything that was in G0, G1, collected everything into a test tube that was in S phase, and separated out everything that was in G2M? What could you do with those cells to then tell you about the cell cycle?
You could look at which proteins were being made during these cycles.
Yes, so you could go back, take those cells, run a Western blot, and see where is cyclin E made, where is cyclin D made. You could do an IP, do a kinase assay. Is cyclin E CDK2 functional in G0, G1? So there's all kinds of things you could find out about doing FACS and cell sorting, okay? All right. Okay, so far we know that from the cell cycle that cyclin D CDK4 will block RB activity, that cyclin E CDK2, its claim to fame is to block RB activity. That way E2F can be, yes?
Sorry.
That's okay. Sure. Okay.
On the third question, I don't think I got all the steps to
Okay, so the steps are here. This is the slide with the steps, right? So here we go. Growth factors such as mitogens, right, are going to cause cyclin D, CDK, or cyclin D transcription to be made. So there's a figure, this figure here that we talked about on last week is the signaling cascade, right? Platelet-derived growth factor binds to its receptor. It's going to either activate AKT signaling cascade or activate the ERK signaling cascade downstream of that. I have an arrow pointing here. You're going to get cyclin D transcribed. So now you have cyclin D transcribed. Cyclin D is transcribed. Cyclin D is going to bind to cyclin D CDK4 partner. It's going to partially phosphorylate RB. That's going to allow E2F to start transcribing genes like cyclin E. And this is through like a positive feedback loop. Cyclin E CDK2 is going to phosphorylate RB even more. So now RB is completely disassociated from E2F. Now E2F is fully active. It's going to activate or transcribe cyclin A. Cyclin A is what gets you into S phase. Okay? Did that help?
Yeah.
Okay. All right. So we just talked about this. Okay. So here's some experimental evidence. I was telling you that what E2F does, and that's to get you transitioning out of G1 into S phase. Out of G1 into S phase. E2F induces transcription of cyclin A and cyclin E. So that's shown here, this particular experiment. Here's your luciferase assay. So that's what this is. What they did in this case is they are looking at, this is going to be, in this case, they put cyclin E promoter and fuse that to luciferase. So that's what they're doing in this particular experiment. So it's a cyclin E promoter fused to luciferase. What they did is they added E2F to cells, right? So all of these cells are expressing the E2F transcription factor. And then they added in increasing concentrations of the cyclin E CDK2 complex. All right, so this is all done in a test tube. So increasing concentrations of cyclin E CDK2 complex. And what you see here is phosphorylation of E2F. So now let me just back up just so that this doesn't get even more convoluted. One of the ways that E2F is activated is through phosphorylation by cyclin E CDK2. So not only does cyclin E CDK2 phosphorylate RB, but it also phosphorylates E2F to help activate it in this instance, and that's what they're showing here. But the important thing to note here is that as you're adding in cyclin E CDK2, because we know that this complex is gonna phosphorylate RB to help E2F be activated, we see an increase in the cyclin E luciferase activity. And that is because E2F is now, because you have a active complex here, E2F is able to be active and transcribe cyclin E. This is called a positive feedback loop. That's what you're seeing here. So cyclin E is actually driving its own expression. Any questions about this? Okay. All right, and then this is what I was saying before, just about the other thing too that cyclin E CDK2 can do is phosphorylate E2F and activate it. All right, now cyclin A. So the important thing about cell cycle, remember we were talking about this idea, it has to keep moving forward. And so because it has to keep moving forward, at some point you have to turn off the activity. So cyclin E, CDK2 can't always be active. At some point it has to stop being active. E2F can't always be transcribing genes. At some point it's gotta stop because you gotta be able to move forward in the cell cycle. So where cyclin E, CDK2 was acting in a positive feedback loop to drive E2F activity, cyclin A, CDK2 complex is actually gonna block E2F activity. So as cyclin A is being made and able to form its complex with CDK2, it's gonna turn around and block this guy so that this guy is no longer functional so that the cell can go ahead and transition into S phase. And that's what's shown here. So some phosphorylations are activating in the case of the cyclin E, CDK2 phosphorylation of E2F, that's an activating phosphorylation. In this case, cyclin A, CDK2, when that complex binds to E2F, it's gonna phosphorylate it, and that's an inhibitory phosphorylation. And so that's what's shown here. They're taking CDK2, cyclin A, and E2F, putting that together, and it's a nice in vitro kinase assay. And they see here in the presence of CDK2, when these guys form a complex, they're gonna phosphorylate E2F. And this is, in this case, this is an inhibitory phosphorylation of E2F to block it, so it's no longer transcribing genes that are important for G1, so now it can get the cell into S phase. So you're stopping this guy's activity. And so I'm gonna show you evidence of that in just a second, but I wanna introduce this technique. It's called a gel shift assay. And what I want you to care about, you don't need to worry about any of the other links, just the ones I have boxed, okay? So this is all you need to care about is the ones I have boxed. So what this does, this is a way for us to see protein-DNA interactions, okay? So what you do is whenever you run a DNA or protein through a gel, for example, it has a certain amount of mobility. But now if they complex, so here, this is the DNA running through the gel, it's got a certain level of mobility, so it ends up all the way here at the bottom of the gel. That's what's shown here. But now if you complex that DNA with the protein, it's not gonna move through the gel as fast. And so that's what's called the shift, the gel shift. So then when you look at that piece of DNA again, so this DNA, these little stars it's radio labeled. So we're able to see it because it's labeled, okay? So that shift, you're gonna see a shift in the DNA mobility on the gel because it's complex with a protein. So this tells us that that protein is actually bound to that DNA. So that's gonna be important because if you imagine this protein could be E2F, the transcription factor, bound to the promoter sequence, the DNA sequence of, say, cyclin E, okay? So you could run in a gel the cyclin E, the promoter sequence for cyclin E. Here it is, it's running through, it has a certain level of mobility. And then you complex it with the E2F transcription factor, it's gonna slow that DNA from running through the gel, it's gonna shift it up. So that tells us that there's an interaction. All right, so that's what's done here in this experiment. So here, they're looking at E2F, they're looking at the DNA element that they're looking at, I think in this case, it's an E2F response element. So it's gonna be a piece of DNA that we know that E2F binds to, it's the promoter sequence that we know E2F transcription factor binds, okay? So that here, in the absence of cyclin A and CDK2, remember this is the guy that's inhibitory to E2F. So if this guy is not around, then E2F has no problem binding to DNA. That's where there's that shift. Now, if you start adding in ATP, which is going to be where you get the phosphate from to phosphorylate E2F, and you add in cyclin A and CDK2 to the mix, now see you have here lower, and now at this point, because they add in more cyclin A, CDK2, no binding. So that's showing that this complex, cyclin A, CDK2, when it phosphorylates E2F, which I showed you before, it's going to prevent E2F from binding to any DNA. And so that's why you no longer have this shift there. Does that make sense? Does that make sense to you guys? Any questions on how this works? Yes? No? Okay, yeah. All right? So that's experimental evidence that we know that cyclin A, CDK2, it's going to phosphorylate E2F and prevent it from binding to DNA. So that's why you lose that shift there. All right. Yes.
So, cyclin E also phosphorylates
Mhm.
E2F, but
In that case, it's activating phosphorylation. So phosphorylations could activate a protein or they can inhibit a protein. In the case of the cyclin E CDK2 complex, it's promoting activity of that E2F. In the case of the cyclin A CDK2 complex, it's promoting inhibition. The other thing too I want to make the point of is one, and this is really important about these cyclins. Notice the CDK2 is the same, right? It's the same kinase. What makes the function of that CDK2 different is the cyclin it's bound to, right? So that's really important to know, right? Because it's a kinase. But what makes its function different is the cyclin it's bound to. So we got the same CDK2, it's phosphorylating E2F in both cases, but depending on the cyclin it's bound to dictates what the outcome is. Yeah, okay, all right. Okay, so now moving on, so let's go back here, let's go through this again. So what's not shown here is you have your mitogens that are gonna get the cell out of G0 into G1. Once you're in G1, cyclin D, CDK4, CDK6, their claim to fame, that complex is that it's blocking RB, so that E2F can start to be active. Cyclin E, CDK2, part of its claim to fame is that it's also blocking RB, and it's activating E2F. And now to get you out of G1 and get you transitioned into S phase, you need cyclin A, CDK2 to now, through a negative feedback loop, block E2F activity. Now, once you're in S phase, this guy's claim to fame is actually DNA replication, right? So remember, that's what's happening in S phase, is the DNA is being replicated. And it's gonna replicate the DNA through these important proteins, CDT1, CDC6, and the ORC complex. So, just to start, when we're replicating our DNA, there's these sequences in the genome called origin of replication sequences, and it tells the cell where to initiate replication. One of the super important things about replication is you want it to happen one time in the cell. Okay, so I'm going to go through that in the next slide. You only want your DNA replicated once, not multiple times. So you're going to have multiple different, because the genome is large, so you need multiple origins of replication. I can't remember how many origins of replication we have in our genome, but there's lots and lots and lots. And so these are sequences where the cell knows to sit down the replication complex of proteins, which I'm going to talk about in a second, and start initiation of replicating your DNA. And again, it needs to only happen one time. So here's the process. Here's your origin of replication. The ORCs are going to bind that ORC complex. The complex of proteins is going to bind to the origin of replication. The protein CDC6 interacts with the ORCs, and it helps direct CDT1, and these proteins here, these are called mini chromosome maintenance proteins. This is a helicase that's going to unwind the DNA, okay? So this CDC6 is going to guide these guys, guide this complex through CDT1, guide it to the origin of replication. That origin of replication has been marked by these ORC proteins, okay? So now, once you've got those helicases sitting down at that ORC site, you've got to get rid of CDC6 and CDT1, because you don't want to start bringing more ORCs, right? You want to just replicate, you don't want to start bringing more helicases. You wanna this process needs to happen one time. So these guys are getting phosphorylated so that they can get off of the complex. So, and what phosphorylates these guys is cyclin A. So, and that happens in S phase, because that's where cyclin A is made, right? Cyclin A is made in S phase, and now it's around to phosphorylate CDT1 and CDC6 so it can get away from this complex. So now you've got your helicases here bound to the ORCs. Now the helicases are gonna start unwinding the DNA, and then you get proteins to come in and replicate the DNA. This is gonna happen, and then the ORCs get phosphorylated and they get removed off of the DNA again so that you don't replicate twice. You only want one replication event per cell cycle, okay? And these phosphorylations, I think, okay, I've got here, right? The ORCs recognize origin of replication. CDT1 and CDC6 are important for recruiting the mini chromosome maintenance complex, recruiting this helicase to the ORC. And then the mini chromosome maintenance and the GEN protein, which is shown here, these are the helicases. They're going to unwind the DNA so that it can start getting replicated. And I show here, I say here, so cyclin A CDK2 is important for phosphorylating CDT1, CDC6 in the ORCs, so you get them off of, once replication is started, you want to get these guys away from the DNA so that the replication only happens once. And then there's a protein, you don't have to worry about this, but this kinase DK phosphorylates the mini chromosome maintenance proteins to activate them. Okay, but the important thing here to note is that S phase, which is where cyclin A CDK2 is functional, that's where its claim to fame is happening, where it's promoting replication of the genome. All right, so I already talked about this. So these guys get phosphorylated by cyclin A CDK2. Experimental evidence of that is going to be through our nice in vitro assay. So let me just go through this real quick. What we're going to see on the next slide is cyclin, I think it's going to probably be cyclin A CDK2. It's going to bind with something called an E3 ubiquitin ligase. Do you guys know what that is? An E3 ubiquitin ligase? So it's something that puts ubiquitins, helps put ubiquitins onto proteins to mark them for degradation. Okay, so cyclin A CDK2 is going to be bound. You're going to mix that with an E3 ubiquitin ligase. In this case, the substrate is going to be our CDT1 or CDC6 that has to get phosphorylated so it can get off of the origin of replication complex, and then you got your ATP that you need to phosphorylate the complex. So you mix all this stuff together. So your cyclin A CDK complex is bound to the substrate. In this case, it's going to be CDT1 or the ORCs or the CDC6. It gets phosphorylated, whatever that substrate might be. And then that substrate is going to be bound to the E3 ubiquitin ligase, and this tells us that downstream of this, it's going to be marked for degradation. The point that I want to make here that's important about this slide is, in this case, and you're going to see it in the next slide, in order for this guy to get marked for degradation, it has to be phosphorylated, right? So the only way, if we go back here to this slide here, in order for this guy to get degraded, this guy to get degraded, this complex to get degraded, in order for these things to happen, they have to be phosphorylated by cyclin A CDK2, okay? And once they're phosphorylated, they're marked for degradation. So once they're phosphorylated, they can bind to the E3 ubiquitin ligase, and that'll get them degraded so that you only go through this process one time during the cell cycle. You don't want to replicate the DNA twice. Okay, so that's what's shown here. Okay, so what they did in this experiment, they have cyclin A CDK2, they IP for CDT1, so they IP for that CDT1, and what they see is in the presence of cyclin A CDK2, that guy's phosphorylated, so that's the shift here on the gel. In the absence of any kinase activity, so if you don't have CDK2 in that complex, this CDT1 is not going to be phosphorylated. And then if you look here, they did an IP, right? So they're IPing for CDT1. They notice, look, that this E3 ubiquitin ligase is called SCIP2. This guy can only bind to CDT1 if it gets phosphorylated by cyclin A CDK2. So that's the only way you can pull down this E3 E3 ubiquitin ligase is if this guy gets phosphorylated by this complex. If cyclin ACDK2 is not active, so you have a kinase here, it doesn't get phosphorylated, right? So you don't see a shift, and you don't see any pulling down of the E3 ubiquitin ligase. So this is experimental evidence is telling us that in order for that substrate, CDT1, to be marked for degradation by this E3 ubiquitin ligase, it has to be phosphorylated by the cyclin CDK complex. And that's the mechanism of how this works. Does that make sense? Okay. Okay, and remember before I was telling you guys the really interesting thing about these cyclin CDK complexes is this activity of the cyclins, or the CDKs is dictated by what cyclin it's bound to. So here, the real active one complex is gonna be cyclin ACDK2 phosphorylating CDT1 so that it can be phosphorylated and marked for degradation so that you don't get replication happening twice. And it's marked for degradation, we know that because it binds through to its E3 ubiquitin ligase. Cyclin A, CDK1 has some level of activity, right? So you can see if cyclin A is bound to CDK1, that can phosphorylate CDT1. We see that in the shift. And there's some binding to the E3 ubiquitin ligase. But now look, if CDK1 is bound to cyclin B, you get no phosphorylation, you get no binding to the E3 ubiquitin ligase. So again, this goes back to the importance of the activity of these CDKs. It's dictated by what cyclin it's bound to. Okay? Okay, and then here is the next experiment is going to be talking about the ORCs. So there's something that we do called a chip assay. Has anybody ever heard of chip? Anybody done chip in the lab? Okay. So the idea behind chip is if we want to show, it's another way of showing experimentally that a transcription factor is bound to a promoter region. So it's a way of showing a protein is bound to a piece of DNA. In this case, we're concerned about a transcription factor binding to, well actually in this case, it's not gonna be a transcription factor, it's gonna be the ORC complex. In this case, we care about the ORC complex being bound to the origin of replication. So the idea here, this is your protein complex here, bound to DNA, okay? So what you do is you fix the cells, you cross-link the cells with something like formaldehyde. So everything that's going on in the cell at that time is fixed in time, fixed in place. So now you've fixed all of these proteins, whatever these proteins might be, bound to the DNA, it's fixed. Then you sonicate or digest these pieces of DNA because you can't, you gotta break the DNA up to be able into manageable pieces. Okay, so here, let's say we really care about whatever this green protein is. So we use an antibody to that green protein because we want to know what pieces of DNA in the genome that green protein is bound to. So we use an antibody to that green protein and do our immunoprecipitation and pull out everything that that green protein is bound to. And so something that's going to be bound to is DNA. So then we do PCR to amplify that piece of DNA because we're not going to be able to see that one piece of DNA. So we have to do PCR to amplify this region of DNA up, and then we can run it on a DNA gel, or you can do qPCR or something and see the signal. So here's what that looks like. So in this case, we're looking specifically at the ORCs. Remember, the ORCs are the complex that actually binds to the origin of replication to tell the cell, this is where I want to start replicating my DNA. Now, remember, we know that cyclin A, CDK2, is the complex that is going to phosphorylate this guy to get him off of the DNA once the replication is started. So that's what's shown here, is in the case of, here we go, so the ORC, your IPing for ORC, this is during G1 phase. So remember, cyclin A is functional during S phase. During G1 phase, cyclin A is not around, right? It's not active. So in G1 phase, the ORC is nice and bound to DNA, and that's what's shown here. So you IP for ORC, that's what you did up here. You're doing a chip assay. So you're gonna amplify the piece of DNA that the ORC is bound to, and that's what you're seeing here in the PCR, okay? Right here, this is a mutant, this is AA, this is some sequence that was changed to to alanines that prevents the ORC from being phosphorylated. So remember, once ORC is phosphorylated, it has to get off of the DNA. Well, if you can't phosphorylate the ORC, it stays stuck to the DNA. So that's what's shown here. And then here, this is a phosphomimetic. So basically what this is an ORC that thinks it's phosphorylated all the time. So because it's thinking it's phosphorylated all the time, it never binds to DNA. And so that's what you're seeing here. That's why you get no band in the chip assay. Okay? So one of the important things here, this is G1 phase where they isolated this protein from these cells. And remember, in G1 phase, you don't have an active cyclin A CDK2 complex, which is why in G1 phase, this guy can be bound to DNA, that ORC complex. Okay? All right. And then here, this is going into G2M phase. So now here, this is not shown here on this slide, but this is after the DNA is replicated. So what they're showing here is that once you hit G2M phase, you can't have this guy bound to the DNA anymore, because remember, you can only do one replication cycle during a cell cycle. So in G2M, it's not bound, and that makes sense. Because you know, in S phase, cyclin A phosphorylates it and gets it off of the DNA. So in the G2M phase, there's no ORC bound to DNA. If you have this guy that can't be phosphorylated, it's going to stay stuck on the DNA, even going into G2M phase. And then this is that phosphomimetic again. This is the guy that thinks it's always phosphorylated, and so of course in G2M it's not bound. Okay, but the important thing to note here in G1 phase, the ORC is bound to the DNA, and once you get to G2M phase, it's not bound. And that goes back to UT Dallas green shirt, what we were talking about, how you can separate things right out into the cell cycle and look at what's going on during different phases of the cell cycle.